The mass increased, which caused osmosis to happen in the Visking tubing. Okay, that exact sentence right there, it is a devastating exam mistake. If you write this, you're totally confusing cause and effect, and honestly, it's going to cost you vital grade boundaries in the literal blink of an eye. Cambridge examiners see this all the time, and they will bring out the red pen within the first 3 seconds of reading it.
Welcome to today's explainer. If you're aiming to absolutely master Cambridge Biology Topic 4.2, you are in exactly the right place. Today, we aren't just reviewing biology, we're cracking open the examiner's playbook. We're uncovering exactly what they demand from you to secure those top band marks. So, let's dive right in.
Section 1. The costly exam mistake, and exactly why ordinary revision fails.
Look, you can memorize every single biological concept, but if you don't know how to navigate the pitfalls reported directly from the Cambridge Principal Examiner reports, you're just leaving points on the table. These are the top 3 mistakes that trip up even the best candidates. First, as we just saw, students constantly confuse cause and effect. Remember, osmosis causes the mass to change, not the other way around. Second, under exam pressure, candidates completely forget to check their units and terminology. And third, they provide generic, memorized answers instead of actually tailoring their response specifically to the context of the Visking tubing or Agar investigation that's right there in the question. It is incredibly easy to lose marks here, even if you know the underlying theory perfectly. Consider this a massive, flashing red light straight from the Principal Examiner reports. Candidates consistently confuse key terminology. My advice? Create your own personal mnemonic for all the key terms in Topic 4.2. Use the first letters of words like water potential, concentration gradient, net movement, and selectively permeable to lock these into your memory. This ensures you never ever mix them up under intense exam pressure, and it forces you to always define your terms clearly before you launch into those extended responses. Moving on to Section 2, the method mark strategy, or winning marks through process. A lot of students think they only get points for arriving at the final correct biological conclusion. But guess what? Examiners heavily reward the logical process of an investigation. To win these method marks, we need a solid step-by-step framework. First up, explicitly state that you'd select appropriate apparatus. Next, follow and describe the sequential instructions. Third, detail exactly how you'd record data in suitable tables, and yeah, that means proper headings and units. Fourth, actively identify any anomalies, and finally, draw evidence-based conclusions. The absolute crucial point here is that this exact logical flow is precisely how the 9700 practical and theory mark schemes are built. If you skip a step in your explanation, well, you skip a mark. But wait, this brings us to a massive question. What is the hidden examiner clue that changes the answer completely? In just a moment, I'm going to reveal a hidden, single-word clue that literally flips the entire mark scheme upside down. It's a trap that catches thousands of students. But before I give you that clue, you need the analytical framework that makes this trap completely obvious to spot.
Section 3. Systematic data analysis. Choosing the right statistical weapon.
I know that interpreting complex data and terrifying graphs is where students panic the most during Topic 4.2 exams, but we have a foolproof system. Take a mental screenshot of this slide right now, because it brilliantly illustrates exactly when to deploy each statistical analysis tool, without second-guessing yourself in the exam hall. If a question asks you to compare the means between 2 sets of data, say 2 different concentrations of sucrose and Visking tubing, you deploy the t-test. If you're analyzing categorical data frequencies, bam, it's the chi-squared test. And if you need to find a relationship between continuous variables, use correlation tests. Memorize this breakdown, and I promise you, you will never pick the wrong statistical weapon again.
Section 4. Spot the flawed answer. Can you beat the examiner? All right, here's a quick pause and predict moment for you. Think about the methodology and terminology we literally just covered. Try to anticipate exactly where a student might drop 3 critical marks when explaining osmosis in an Agar block or a Visking tubing experiment. You have it? Let's see if you're right.
Imagine holding a red examiner's pen right now. A phrase like mass change so osmosis happened in the Agar is a flawed, vague, cause and effect disaster. It earns 0 marks. 0. Now, compare that to the top band scientific reasoning on the right side of the screen. Water potential gradient caused net water movement into the Visking tubing, increasing mass. This is perfect. It defines the mechanism, the water potential gradient. It describes the precise action, net water movement, and it states the correct outcome, increasing mass.
Section 5. Writing the perfect answer. The Deconstruct, Solve, Evaluate framework.
Now that you see the difference, here's the ultimate strategy for writing that perfect answer every single time. This sequence right here is a high-retention system. Top-scoring candidates use this exact framework to break down massive walls of stimulus material into manageable, mark-winning chunks. Step 1. Deconstruct the question and extract the raw data. Step 2. Select your analytical framework, just like we did with the stats table earlier. Step 3. Solve the problem systematically, showing absolutely all you're working. And Step 4. Evaluate your answer, to check if the biological outcome is actually, reasonable.
And when that evaluation requires comparative analysis, merely listing isolated facts just won't cut it. You must identify both similarities and differences. You've got to use explicit comparative connectives, words like whereas or conversely. You have to support every single comparison with hard numerical evidence extracted directly from the text or graph. Finally, draw powerful conclusions about their relative importance. You absolutely must question assumptions and justify your decisions.
Section 6. The hidden mark killer, the unseen trap.
Okay, we've covered the biological theory, the practical steps, and the analytical frameworks. But now it is time to pivot to the final, most crucial hurdle. The biological science was perfect, but the command word was ignored. This is the diagnostic cliffhanger of our entire explainer. Students are losing entire mark bands, literally dropping from an A to a B or worse, not because they don't understand the complex biology of Visking tubing, but because they completely misinterpret what the examiner is fundamentally asking them to do.
Ask yourself, does describe mean the exact same thing as explain? Think about it. If you answer an explain question, which requires you to give scientific reasons and actual biological mechanisms with a describe response, which basically just asks you to state what you see on a graph, it is a catastrophic error. You will cap your grade immediately, no matter how incredibly accurate your description is. You desperately need to know how to decode these commands to unlock that top tier of marks. The exact solution to that critical problem comes down to mastering those command words. But it leaves us with a much bigger question. If the difference between an A and a B comes down to a single word like describe versus explain, are these exams truly testing our biological knowledge, or are they just testing our ability to decode a linguistic puzzle? Think about that as you head into your next revision session. And to make sure you're always on the winning side of that puzzle, hit subscribe to our YouTube channel Ace the Exams at acetheexams_IGCS to excel in your Cambridge examinations, and click the next video right now to master command words.